Hello everyone, this is Latha Akula. In this video, we are going to study about the next phase in interphase, that is the S phase. Already in the last video, we have completed the G1 phase. What happens in the G1 phase? There is the size of the DNA increases and also the components required for the S phase will be prepared in the G1 phase only. There is no DNA replication occurs in the G1 phase. Then where the DNA replication occurs? In the S phase. So here what is S phase? It is a synthesis phase. It is synthesis phase. What it's synthesizing? DNA. Here in this S phase replication of DNA occurs. It is synthesis of DNA occurs and hence we are calling it as a S phase. And if you see, in the cell cycle, G1, G2, M phase, in all these, S phase is the longest phase of all these cell cycle. What is the longest phase? S phase. Here, the replication of DNA occurs and here the cell doubles the amount of DNA. What happens here? The amount of DNA gets doubled. For example, the initial amount of the DNA in the cell is 2C and after S phase, the amount of DNA doubles. That means 2, what is the doubling of 2? 4. That means 2C becomes 4C. Here the C is nothing but the amount of DNA is represented by the C value. Here, amount of DNA doubles, but there is no change in the number of chromosomes. We need to remind it. So what happens in the S phase? Only the amount of DNA doubles, but there is no change in the number of chromosomes. That means the chromosomes remain the same. There is no change occurs here. For example, in the diploid cell, we have two types of cells, haploid cells and diploid cells. Diploid cells means the cells with two of its chromosomes is called as diploid cell, and haploid cell means the cell with one of its chromosome is called as haploid cells. Diploid condition is denoted by the 2n and haploid condition is denoted by the n. For example, diploid cell, it has 2n chromosomes in the G1 phase. After S phase, the number of chromosomes will be 2n only. There is no change in the number of chromosomes. Whereas in haploid cells, the initial number of chromosomes was n and after S phase, the number of chromosomes will be n only. There is no change in the number of chromosomes. Here, the existing DNA, this is DNA. The DNA is a double helical structure. It has two strands. So during replication, the DNA gets opened and each strand will act as a template. So each strand synthesizes the new DNA molecule. This is a new DNA molecule. This is a new DNA molecules. So these are the two DNA molecules produced from the one DNA. Here, the existing DNA molecule will act as a template and synthesizes the new DNA molecules. Here, if you see in the cell, before cellulation, what happens? In the nucleus, the genetic material was in the form of a thread-like structure. So, this thread-like structure, what we call it as chromatin. This chromatin, during replication, during cellulation, what will happen? This chromatin, it gets condensed, it gets coiled around histone proteins and form a structure called as chromosome. Chromatin changes into condenses and form a structure. It form a definite structure called as before chromosome. The chromatin it don't have any definite structure. So during cell division, what happens? The chromatin condenses and form a definite structure called as chromosome. So this is the chromosome. So before replication, the chromosome structure will be like this. This is also chromosome only. But before replication, the structure of the chromosome will be like this. This is called as chromatid. At the center, the chromatid has only one arm. One arm of the chromosome is called as chromatid. At the center, it has centromere. After replication, the chromosome, it makes its copy and forms two chromatids. We can call these two chromatids as the sister chromatids also. The two sister chromatids, they join by attached together by means of centromere. Here there is no change in the number of chromosome. So only one centromere here, here also only one centromere. Here one chromosome, this is also one chromosome only. So only change in the number of chromatids. Here it has one chromatid. After replication, it becomes two chromatids. This is also a single chromosome. This is also a chromosome only. But here, the number of chromatids only changes. That means there is no change in the number of chromosomes. Only amount of DNA increases. Amount of DNA doubles. That is what we are discussing about. Here, animal cells, in the animal cell, the DNA replication, where the DNA replication begins in the nucleus, and at the same time above the nucleus there is a structure called as centriole. The centrioles, duplication of the centrioles occurs in the cytoplasm. This is for example, this is the cell. The center there is a nucleus. Within the nucleus there is a chromatin, right? Above this nucleus there is a structure called as centrioles. These centrioles, they are helpful in the M phase. During M phase, these centrioles, they play major role. And here the duplication of these centrioles also occurs in the cytoplasm, and these centrioles were absent in the plant cell. So this is about the S phase. What happens here? Amount of DNA doubles. There is no change in the number of chromosomes. And what happens in the animal cells? The DNA replication occurs in the nucleus, and the centrioles duplication occurs in the cytoplasm. And this is about S phase. And the next one is the G2 phase. The next phase is G2 phase. The G2 phase is otherwise called as gap 2 phase. The G1 phase is called as gap 1 phase, and the G2 phase is called as gap 2 phase. And here this G2 phase is the intermediate between S phase and M phase. What happens in this G2 phase? Here the tubulin protein secretion takes place. What is a tubulin protein? What is its function? We see, we have studied in the S phase, there is a duplication of centrioles occurs in the cytoplasm. In that point, what is the importance of that centriole? From the centrioles, the spindle fibers were developed to attach the chromosomes. The spindle fibers are thread-like structures are formed by this tubulin protein. So for this tubulin protein is required in the next M phase, right? For that, before only the tubulin protein was ready. Next, mitochondria, chloroplast and Golgi apparatus duplication occurs here. Here the DNA synthesis stops, but RNA and protein synthesis still continues. Those RNA and proteins are required for the next phase, that is M phase. Here in animals, the mitotic division occurs in the diploid somatic cells. So where it occurs? In the diploid somatic cells. Meiotic division that occurs in the gametes. Sex cells are the gametes, right? In the plant cells, the mitotic division occurs both in haploid and diploid cells. So this is about G2 phase. The next phase is M phase. In the next video, we are going to study about M phase.